Hi, this is Maggie Virus, and this is my Exam 11 case study on Lyme neuroborreliosis. So this slide basically introduces the nervous system. It includes the central nervous system and the peripheral nervous system, and it is involved in sensing the environment, integrating information, and controlling motor responses. So the central nervous system is protected structurally by the skull and vertebrae, and by also meninges, which includes the dura mater, arachnoid matter, and the pia mater, and cerebral spinal fluid, which cushions the brain and the spinal cord. And despite these protections, pathogens like Borrelia burgdorferi can sometimes cross the blood-brain barrier, and a specialized interface that normally restricts microorganisms from entering the CNS. But the CNS's immunologically privileged status limits normal immune responses, so when Lyme's disease affects the nervous system, it can result in meningitis, cranial neuropathies, radiculitis, and basically defining Lyme neuroborreliosis. And this sets the stage for understanding how the bacterium interacts with the nervous system. So now I'm going to talk about the background of this disease. So it's not just a tick-borne infection. It can target the nervous system and lead to serious neurological complications if it goes untreated. And its occurrence is tied to specific geographical regions, mostly regions with high tick prevalence. So public health awareness and preventative measures are key. But historically, the discovery of Lyme disease occurred in Connecticut during the 1970s, and basically it illustrated how epidemiological investigation can reveal a previously unrecognized infectious agent. And Lyme neuroborreliosis exemplifies the complicated interactions between a pathogen and the nervous system, and it shows why this disease remains important focus for research and clinical practices. So this is the main focus of our study, is how Borrelia burgdorferi invades and affects the nervous system. So unlike many bacterial infections, this pathogen must overcome structural defenses like the meninges, the blood-brain barrier, and also navigate the CNS's immunologically privileged status. So basically, I'm just going to explore how the bacterium's immune evasion allows it to persist, and how this interaction leads to neurological manifestations, which is seen in Lyme neuroborreliosis. So it's important to understand these mechanisms for developing effective therapies and informing public health strategies. So this basically sets up the rest of the PowerPoint. So this slide talks about the taxonomy, cell type, structure of the bacteria. So it's a prokaryotic, gram-negative spirochete, and this means it has a helical shape and a thin cell wall. And its endoflagella, located between the inner and outer membranes, give it a corkscrew motility and the extracellular matrix during infection. Its corkscrew motility is crucial for penetrating tissues and the extracellular matrix during infection. But unlike many bacteria, it lacks a capsule and endospores, but it expresses outer surface proteins, which are important for attachment to the host cells and evasion of the immune system. So also it has a flexible structure, which allows it to navigate through connective tissues like the bloodstream and the nervous system, and it sets the stage for the neurological manifestations of this virus. So basically I'm talking about how Borrelia burgdorferi survives and causes diseases. So it's microaerophilic, meaning it prefers environments with low oxygen, and it's also a heterotroph, which relies on host-derived nutrients rather than synthesizing its own. And these metabolic traits allow it to persist in both tick vectors and mammals. So its virulence is largely driven by outer surface proteins that mediate adhesion to host tissues, and antigenic variation, helping it evade the immune system. Proteases are helpful in degrading the extracellular matrix and allow for dissemination into connective tissues and nervous tissues. So genetically, it possesses a linear DNA chromosome and multiple plasmids, which encode for many of its virulence factors and makes it highly adaptable during infection. So here's an overview of Lyme neuroborreliosis. So it primarily affects the nervous system, both central and peripheral. We knew that already. But the CNS involvement can include meningitis, encephalitis, and inflammation of the spinal cord, while the PNS can show nerve inflammation, leading to radiculopathy or cranial nerve dysfunction. And these signs and symptoms are diverse. Early on, patients may experience headache, neck stiffness, fatigue, and fever, which are similar to other infectious diseases. But then as the infection progresses, neurological symptoms will appear, such as facial palsy or radicular pain and peripheral neuropathy. So cognitive symptoms like memory issues and concentration problems may occur, which reflect the pathogen's ability to invade nervous tissues. And then you also may experience sleep and mood disturbances, and these can affect the patient's quality of life. So the incubation period is variable and typically from 1 to 2 weeks after a tick bite, but it can extend into months as the bacterium slowly disseminates through tissues. So the symptomatic duration also varies, with proper treatment and symptoms may resolve in a few weeks, but in some patients, neurological symptoms can persist for months. So complications may cause persistent meningitis or encephalitis, which leads to ongoing inflammation of the CNS. And the peripheral nervous system's involvement can result in neuropathy or radiculitis, while cognitive mood disturbances may persist even after bacterial clearance. So early antibiotic therapy is critical to prevent these outcomes, and most patients recover fully if they're treated quickly. So now I'm going to talk about the transmission and epidemiology. So Lyme neuroborreliosis relies on a vector-host cycle to persist in nature. So the bacteria primarily lives in small mammals, especially white-footed mice, which act as natural reservoirs. Deer are also essential for maintaining adult tick populations, but they do not typically harbor the bacteria themselves. And though infection spreads to humans through tick bites, making Lyme disease a classic vector-borne disease. The risk of transmission increases if the tick remains attached for over 24 hours, about 36 to 48 hours. And this allows the bacteria to migrate from the tick's gut to the host. But human-to-human transmission is essentially negligible, which reinforces the importance of tick avoidance and early removal. So the primary portal of entry for Lyme neuroborreliosis is the skin. So through the infection of the Ixodes tick, humans are considered dead-end hosts. So the bacteria do not typically exit the human body to infect other humans. And susceptibility is highly tied to geography and exposure risk. So people in northeastern and upper Midwestern U.S., as well as parts of Europe, have the greatest risk. And this is also increased if you are engaging in outdoor activities or gardening, which can further increase your exposure. And all children or all ages can be affected, but children and older adults tend to show higher incidences of obtaining neuroborreliosis. So this disease exhibits seasonality and peaks when the nymphal ticks are most active. And recently, climate change has been expanding tick habitats. Tick habitats have contributed to a rising number of reported cases which emphasizes the public health relevance of Lyme disease. So now I'm going to go into the pathogenesis. So the entry of Borrelia burgdorferi begins with the bite of an infected tick. So the bacteria are deposited into the skin along with tick saliva, which contains proteins that will modulate host immunity. And this immunosuppression helps spirochetes avoid early detection by the innate immune system. So once they are in the dermis, the spirochetes will navigate the extracellular matrix and use their adhesion molecules and host proteases to allow them to penetrate deeper into tissue. And the initial immune response is dampened, which gives the bacteria a critical head start for dissemination. And this early phases set up the stage for later neurological involvement for if the bacteria are able to reach the CNS. So now I'm going to talk about attachment. Attachment is critical for Borrelia burgdorferi to establish infection, and the bacteria expresses outer surface proteins that specifically bind components of the extracellular matrix and allows them to adhere to tissues such as the skin and later the CNS. So in Lyme neuroborreliosis, the spirochetes will exploit these adhesion mechanisms to cross the blood-brain barrier, which is normally highly selective. But by attaching to endothelial cells and ECM components, the bacteria reduce immune detection and set up localized infections in the meninges and cerebrospinal fluid. So once Borrelia enters the tissues, it employs multiple strategies to evade the immune system. So the bacteria frequently alter the VLSC surface protein, which makes it difficult for the host to mount an effective antibody response. And it also recruits host complement proteins, and this prevents lysis. And in the CNS, immune evasion is enhanced because microglia are less responsive and adaptive immune cells are slower to arrive. So some spirochetes may even hide inside cells or form biofilms, which further reduces detection and contributes to persistent infection. So now I'm going to talk about damage and response. The primary damage in Lyme neuroborreliosis results from the host immune responses to the bacteria. So cytokine release leads to meningeal inflammation and nerve irritation and sometimes long-term CNS effects. And early neuroborreliosis shows acute signs like facial nerve palsy or meningitis, whereas the late stages include cognitive and peripheral nervous system defects. So the immune system is a double-edged sword, basically. It helps contain the infection, but it also causes much of the tissue damage and neurological symptoms. And finally, I'm going to talk about the exit pathways. So Borrelia burgdorferi does not exit humans to infect others under normal conditions, making it humans a dead-end host. So the natural life cycle continues in ticks, which acquire the bacteria from other rodents. And persistence in the CNS allows for the bacteria to maintain infection over months or years and often causes long-term neurological complications. So this is facilitated by antigenic variation and immune evasion, which is particularly in immunologically privileged sites like the brain and the CSF. And here are my slides. I'm sorry I couldn't pronounce borreliosis every time. Thank you.